Okay, ready to dive into the world of CRISPR.
Absolutely. It's a game changer, no doubt.
CRISPR-Cas9, gene editing, it seems like it's everywhere these days, but what's all the hype about?
Well, for starters, this technology is letting us rewrite the code of life itself. We're talking about potentially curing diseases that were once thought to be incurable.
That's a bold claim. Are we talking about science fiction here, or is this really happening?
It's happening right now, and the science behind it is fascinating. Believe it or not, we have bacteria to thank for this revolutionary technology.
Bacteria. Really? Those microscopic things are giving us the key to editing genes.
It sounds crazy, but it's true. Scientists discovered that bacteria use a system called CRISPR-Cas9 to defend themselves against viruses. It's like their own personal immune system.
So how do bacteria, these tiny organisms, become experts in gene editing? I'm struggling to connect the dots.
Okay, so imagine this. A virus invades a bacterium. The bacterium, using its CRISPR system, takes a mugshot of the virus's DNA and stores it in its own genetic memory bank.
Like a wanted poster for bad viruses.
Exactly. And the next time that virus tries to invade, the bacterium recognizes it instantly. And that's where Cas9 comes in. It acts like a pair of molecular scissors.
Molecular scissors. Okay, now you've got my attention. What do these scissors do?
They're guided by that stored DNA mugshot to precisely snip the invading viral DNA, effectively destroying the threat.
So it's like the Cas9 scissors are the enforcers taking out the bad guys based on the CRISPR memory bank.
Precisely. It's a brilliant defense mechanism. And the truly groundbreaking part is that scientists figured out how to adapt this bacterial system to edit genes.
So we're taking a page from bacteria's playbook to potentially cure diseases.
And not just potentially. We're seeing promising results in treating genetic diseases like cystic fibrosis and sickle cell anemia. This is real.
That's incredible. But how does this bacterial system actually work in, say, a human cell?
Think of it like this. DNA is like a massive library with thousands of books, and each book is a gene. What CRISPR allows us to do is go into that library and target a specific book or gene with incredible accuracy.
So we're not just randomly flipping through pages, we're going straight to the source of the problem.
You got it. And once we've targeted the right gene, we can use those Cas9 scissors to make changes. It could be anything from correcting a single typo in the DNA sequence to inserting an entirely new gene.
It sounds a bit like we're playing molecular surgeons here, going in and making precise incisions in our DNA. But how do we tell those Cas9 scissors exactly where to cut?
That's where guide RNA comes in. Think of it like a GPS navigation system for those Cas9 scissors.
So scientists program the GPS coordinates, which are actually a specific DNA sequence, and then Cas9 just follows the directions.
Exactly. Scientists design a guide RNA molecule that matches the DNA sequence they want to target. Cas9 attaches to this guide RNA, and together they search the DNA for a perfect match.
And when they find that match?
Snip. Cas9 makes a clean break in the DNA strand.
Okay, we've made a cut. What happens next? Does the cell just panic?
Not at all. Cells are remarkably good at repairing themselves. When a DNA strand breaks, the cell activates its own repair mechanisms. It rushes in to fix the damage.
Okay, so the cell is trying to fix the cut, but how does that actually lead to, say, curing a disease?
That's where things get really interesting. There are a couple of ways scientists can use this repair process to our advantage. One way is to simply disrupt a problematic gene.
Disrupt? What do you mean?
So sometimes a gene can be faulty and cause disease. By cutting the DNA at a precise location within that gene, we can essentially make it inactive.
We're essentially turning off the faulty gene, like flipping a switch.
You got it. And without that faulty gene doing its thing, the disease can often be stopped in its tracks. That's the principle behind gene knockout.
Okay, that makes sense for getting rid of a bad gene. But what about when we need to introduce a new gene, like in gene therapy?
That's where things get even more impressive. We can actually use the cell's own repair machinery to insert a new gene. It's a bit like providing the cell with a patch kit along with the scissors.
So we're not just cutting and disrupting, we're also cutting and pasting.
Exactly. We provide the cell with a template of the desired DNA sequence along with the CRISPR-Cas9 system. The cell then uses this template to repair the cut, effectively inserting the new gene into its DNA.
Wow, it's incredible how we're harnessing the power of these cellular processes for gene editing. So what are some of the diseases that scientists are targeting with this technology?
Well, we already mentioned cystic fibrosis and sickle cell anemia, which are caused by single gene mutations. But the potential goes far beyond that. Researchers are exploring CRISPR therapies for everything from cancer and HIV to inherited blindness and muscular dystrophy.
That's a pretty impressive range. It seems like CRISPR is poised to revolutionize medicine as we know it.
Absolutely. And it's not just limited to treating diseases. CRISPR is also being explored for its potential in diagnostics, agriculture, and even biomanufacturing.
This is truly revolutionary stuff. We're talking about potentially reshaping our relationship with biology itself. It really does feel like we're on the verge of a new era in medicine. But with a technology as powerful as CRISPR, I imagine there are also some concerns.
Absolutely. And one of the biggest ones is the potential for off-target effects. Think of it like this. You're trying to make a precise cut in a piece of paper, but your scissors slip a little.
And you end up with a cut in the wrong place. Not ideal, especially when we're talking about DNA. So what are scientists doing to address these off-target
Right.
effects?
Well, they're constantly refining the CRISPR system, making it more precise. One really exciting development is the emergence of what we call base editing.
Base editing. Sounds intriguing. Tell me more. What is it?
Remember how we talked about CRISPR being like molecular scissors? Well, base editing is more like using a pencil and eraser. It allows us to change a single letter in the DNA code without even cutting the
Yeah.
DNA strand.
That sounds way more precise. Less chance of those molecular scissors slipping. So instead of making a big cut, we're just fixing a typo.
Exactly. It's incredibly elegant and much less invasive for the cell. And then there's prime editing, which takes things to a whole new level.
Okay, you've piqued my curiosity. What's so special about prime editing?
Think of prime editing as the ultimate molecular word processor. It allows us to insert or delete entire sequences of DNA, not just single letters.
So if base editing is like fixing typos, prime editing is like rewriting entire paragraphs of the genetic code.
You got it.
Oh.
It's incredibly powerful, and it opens up even more possibilities for precise gene editing.
It's mind-blowing. It seems like we're constantly pushing the boundaries of what's possible with CRISPR. But with all this talk about cutting and pasting DNA, I have to ask, are there any unintended consequences we should be worried about?
That's the million-dollar question, isn't it? And the honest answer is, we're still learning. The field is so new, and we're constantly discovering more about the complexities of the genome.
So how do we balance the potential benefits of CRISPR with the unknowns?
That's where rigorous research and ethical considerations come in. Scientists are very aware of the potential risks, and they're working hard to identify and mitigate them.
So it's about proceeding with caution and carefully evaluating the potential risks and benefits.
Precisely. And it's not just up to scientists. It's a conversation that needs to involve everyone. The decisions we make about CRISPR technology will have far-reaching implications for generations to come.
Absolutely. It's a collective responsibility. So on that note, let's shift gears a bit and talk about something a bit less, well, heavy. I read that CRISPR is being explored in some fascinating ways beyond treating diseases.
Oh, absolutely. The applications for CRISPR are incredibly diverse. One area where we're seeing a lot of excitement is in agriculture.
Agriculture, like genetically modified crops, that's been around for a while. What's CRISPR bringing to the table?
You're right, GMOs are not new, but CRISPR offers a level of precision and efficiency that's unprecedented. We're talking about developing crops that are more resistant to pests, diseases, and even droughts.
That sounds incredibly promising, especially with the growing concerns about food security and climate change.
Exactly. Imagine a world where we can grow crops that require less water, are less susceptible to disease, and can withstand harsher environmental conditions. CRISPR has the potential to transform agriculture as we know it.
It's amazing to think about. And it's not just about food, is it? I read that scientists are also using CRISPR to develop new biofuels and sustainable materials.
That's right. CRISPR is being used to engineer microorganisms that can produce biofuels, bioplastics, and other materials in a more sustainable and efficient way. It's a completely new approach to manufacturing.
Wow, we really are talking about a potential revolution in multiple industries. From medicine and agriculture to manufacturing and beyond, CRISPR seems to be popping up everywhere. But with all this potential, I have to ask, are there any downsides to using CRISPR in these non-medical applications?
Just like with any new technology, there are always potential risks and ethical considerations. For example, some people are concerned about the environmental impact of releasing genetically modified organisms into the wild.
That's a valid concern. How do we ensure that CRISPR is used responsibly and ethically, not just in medicine, but in all these other areas as well?
It's all about having open and transparent conversations, establishing clear guidelines and regulations, and proceeding with caution. This technology has immense potential for good, but it's crucial that we use it wisely and responsibly.
Well said. It's clear that CRISPR is a powerful tool, and like any tool, it can be used for good or for ill. It's our responsibility to ensure that its potential is harnessed for the benefit of humanity and our planet. Welcome back to the deep dive, where we're wrapping up our exploration of CRISPR. I have to admit, my mind is still buzzing from our last conversation.
It's a lot to take in, that's for sure. But it's not every day we get to talk about something as groundbreaking as rewriting the code of life, right?
You got that right. So, where do we even go from here? What's next for CRISPR? Are we talking flying cars and teleportation next? Maybe not quite yet, but what are you most excited about?
Well, while we're not teleporting just yet, CRISPR is already making waves in some pretty incredible areas. One that I'm particularly excited about is diagnostics.
Diagnostics. Okay, so not teleportation, but helping us detect diseases earlier. That's still pretty huge. Tell me more.
Imagine a world where we can detect diseases like cancer, HIV, or even genetic disorders with a simple, inexpensive test, even before any symptoms appear. That's the kind of future CRISPR diagnostics could unlock.
That would be game-changing. No more invasive procedures or waiting for symptoms to develop. We could catch diseases early on when treatment is most effective. It's like having a crystal ball for our health.
It's pretty close. And the best part is that CRISPR-based diagnostics have the potential to be incredibly accurate, fast, and affordable, which means they could be deployed even in resource-limited settings. We're talking about potentially revolutionizing healthcare on a global scale.
That's huge. It's incredible to think about the impact this could have. So CRISPR is moving beyond just treating diseases and into helping us prevent them altogether. What other surprises does CRISPR have in store for us? What other fields are being impacted by this technology?
Well, we've already touched on agriculture, but that's just the tip of the iceberg. Scientists are using CRISPR to develop crops that are not only more resistant to pests and diseases, but also to drought and harsh environmental conditions.
So CRISPR could help us address some of the biggest challenges facing agriculture today, especially with climate change threatening food security around the world.
Exactly. Imagine crops that require less water, less pesticides, and can thrive in even the most challenging environments. That's the kind of future CRISPR could help us build.
It's like giving our crops a superpower. And all this from a technology that was discovered in bacteria. It's amazing to me how something so small can lead to such massive changes.
It really speaks to the power of scientific curiosity, doesn't it? And here's another mind-blowing thought. CRISPR isn't just changing how we treat diseases or grow food, it's also changing how we understand life itself.
Okay, now you're really blowing my mind here. How is CRISPR transforming our understanding of life? That seems like a huge statement.
Researchers are using CRISPR to explore the very building blocks of life, to study the function of genes with incredible precision, to unlock the secrets hidden within our DNA. It's like having a powerful new lens that allows us to see the intricate workings of biology in a way we never could before.
That's incredible. It's like we're entering a whole new era of biological exploration, all thanks to this amazing tool. But with all this talk about the incredible potential of CRISPR, I think it's important to remind ourselves that it's not without its challenges, right?
You're absolutely right. CRISPR is a powerful tool, but it's not perfect. We've already talked about off-target effects, and those are still a concern. Scientists are constantly working to improve the precision and safety of CRISPR, but it's an ongoing process.
So while the possibilities are incredibly exciting, it's important to approach this technology with a healthy dose of caution. What are some other challenges that researchers are grappling with?
One of the biggest challenges is delivery. How to get the CRISPR system to the right cells in the body safely and efficiently. It's a bit like trying to deliver a tiny package to a specific address in a bustling city.
It sounds like we need a really good delivery system, one that can navigate the complexities of our biology and deliver the CRISPR cargo precisely where it needs to go.
Exactly. And scientists are exploring a variety of approaches, from using nanoparticles as delivery vehicles to modifying viruses to carry the CRISPR system. It's an active area of research, and there's a lot of progress being made.
That's good to hear. So as we wrap up our deep dive into the world of CRISPR, what's the main message you want our listeners to walk away with?
CRISPR is a revolutionary technology with the potential to reshape our world in profound ways. But it's up to all of us to ensure that it's used responsibly, ethically, and for the benefit of all humankind. Stay informed, ask questions, and be part of the conversation.
Beautifully said. It's a future worth shaping together. A huge thank you to our expert for joining us on this incredible journey into the world of CRISPR. And to our listeners, thank you for joining us on the deep dive. We'll see you next time when we'll dive into another fascinating topic shaping our world. Until then, stay curious.